Welcome to this lesson on DNA fingerprinting, a powerful molecular technique used for genetic identification. DNA fingerprinting has revolutionized fields like forensic science, paternity testing, and medical diagnostics. Throughout this video, we'll explore how scientists can identify individuals based on their unique genetic patterns. Let's dive into the fascinating world of DNA analysis and discover how this technology works. DNA fingerprinting is a molecular technique that analyzes unique patterns in an individual's DNA to establish identity. Think of it as a genetic barcode that is specific to each person. Just like your physical fingerprints are unique to you, your DNA contains distinct patterns that can be used to identify you. This technique examines specific regions of DNA that vary greatly between individuals, making it an extremely reliable method for identification purposes. The foundation of DNA fingerprinting lies in an important principle about human genetics. All humans share approximately 99.9% DNA similarity. This means that except for identical twins who have identical DNA, the remaining 0.1% difference is what makes each person genetically unique. This small but significant variation is enough to distinguish one individual from another. And it's this difference that DNA fingerprinting exploits for identification purposes. The key to DNA fingerprinting lies in structures called VNTRs, which stands for Variable Number Tandem Repeats. These are short DNA sequences that are repeated multiple times in a row at specific locations in our genome. Imagine a sequence like ATG repeated several times consecutively. These repetitive sequences don't code for proteins, but they serve as excellent markers for identification because the number of repeats varies significantly between individuals. Let me illustrate how VNTRs vary between people. Person A might have the sequence ATG repeated 4 times at a particular location in their DNA. Meanwhile, person B might have the same ATG sequence repeated 6 times at that exact same location. This difference in the number of repeats creates a unique pattern for each individual. Scientists analyze multiple VNTR locations simultaneously, creating a distinctive genetic profile that is virtually unique to each person. DNA fingerprinting follows a systematic 4-step process. Step 1 is DNA extraction, where DNA is isolated from a biological sample such as blood, saliva, or hair. Step 2 involves restriction digestion, where special enzymes cut the DNA into fragments. Step 3 is gel electrophoresis, which separates these DNA fragments by size. Finally, Step 4 is pattern analysis, where scientists examine the unique banding pattern created to identify the individual. Each step is crucial for obtaining accurate results. Restriction enzymes are often called molecular scissors because they cut DNA at specific recognition sites. These enzymes are naturally found in bacteria, where they serve as a defense mechanism. In DNA fingerprinting, restriction enzymes are used strategically to cut DNA into fragments of varying lengths. Since individuals have different VNTR patterns, the restriction enzymes will cut at different positions, producing fragments of different sizes for each person. This creates the unique pattern we need for identification. Gel electrophoresis is the technique used to separate DNA fragments by size using an electric field. The DNA fragments are loaded into a gel matrix and an electric current is applied. Since DNA is negatively charged, it moves toward the positive electrode. Here's the key principle. Smaller fragments move faster and travel farther through the gel, while larger fragments move more slowly and don't travel as far. This creates a pattern of bands, with each band representing DNA fragments of a particular size. DNA fingerprinting has numerous practical applications across different fields. In forensic science, it's used for criminal investigations and analyzing evidence from crime scenes. For paternity testing, it helps establish biological relationships between parents and children. In medical genetics, DNA fingerprinting assists in disease diagnosis and identifying genetic disorders. It's also valuable in research, particularly in evolutionary biology and population studies. The versatility of this technique makes it an indispensable tool in modern science and medicine. Let's recap the most important concepts from this lesson. First, DNA fingerprinting analyzes unique VNTR patterns in individual genomes to establish identity. Second, the technique uses restriction enzymes to cut DNA and gel electrophoresis to separate fragments, creating distinct band patterns that are unique to each person. Finally, DNA fingerprinting is widely applied in forensics, paternity testing, and medical genetics, making it one of the most important biotechnology tools available today. Understanding these principles gives you insight into how modern science identifies individuals at the molecular level.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.